All right. Hi, everyone, and welcome to the virtual symposium for this afternoon session, Soil Organic Matter, Building a Foundation for Agroecological Transition with Joel Williams. Bienvenue tout le monde au Symposium Sol Vivant virtuel pour la session de cet après-midi, Matière organique du sol, jeter les bases de la transition agroécologique avec Joel Williams. So just before we get started, I'd like to explain a few technical details to make sure this session goes smoothly for everyone. So today's session will be in English with an awesome Australian accent on top of that. And we're happy to be offering live simultaneous translation for all those who would like to listen to the session in French. So I'll be saying a few words in French now just to explain how the interpretation tool works. Alors, je disais qu'aujourd'hui, nous offrons la traduction simultanée pour la session qui est en anglais. Donc, pour écouter en français, vous pouvez sélectionner l'icône d'interprétation en bas de votre écran et sélectionner la langue française. Donc, nos traducteurs vous guideront en français à travers la session. Merci à nos traducteurs. Ça m'impressionne à chaque fois là, de voir à quel point ils sont talentueux. Comment ils travaillent rapidement. Donc, juste pour mentionner, c'est ça, il y a certaines personnes qui semblaient avoir quelques petits problèmes techniques lors des sessions précédentes avec l'outil de traduction. Donc, il faut bien s'assurer que votre Zoom est mis à jour pour bien entendre la traduction. Donc, si jamais vous avez besoin d'assistance ou que ça ne semble pas fonctionner, essayez peut-être de mettre à jour votre Zoom. Et sinon, si ça ne fonctionne toujours pas, écrivez-nous dans le chat avec l'icône converser là, en bas de l'écran. Puis, on va essayer de vous répondre rapidement. So I'll switch back to English now. Everyone should be able to understand. Can I get a few raised hands, actually, just to see who is using the interpretation tool? So to raise your hand, you can click at the bottom of your screen, the hand icon. Wonderful. Okay, a lot of you are using it. Great, thank you so much. You can put your hands back down. Thank you. All right, great. So everybody should be able to understand now. So I'm Sarah from Regeneration Canada. I'm here with my colleagues Antonius and Gabrielle. You guys want to say hi?
Hi, everyone.
Hi, everyone.
And Jael is with us also in the back end helping us make sure everything goes smoothly today. So again, if you have any technical questions, please write them in the chat and we'll get back to you as soon as possible to help you watch the session. So I'm just going to change slides here. There we go. We have 90 minutes together today. And just to make things a little bit more interactive, we're going to be doing two short Q&A periods. So please write your questions in the Q&A using the Q&A tool. So it's the icon at the bottom of your screen that says Q&A. And after the first 35 to 40 minutes, Joel will be taking a few questions. And then for the second part of the presentation, we'll do another short Q&A at the end. So I'd just like to point out too that the first part of Joel's presentation will be more scientific and technical, and the second part is going to be more focused on practices. So you can save your more practical questions for the second part. So just to recap, technical questions you can ask us via the chat, and all your questions for Joel, you can ask them directly in the Q&A. And you can also vote for other participants' questions by giving their question a thumbs up, and we'll get to the more popular questions quicker. So just before I get started with introducing Joel, I'd like to thank some of our event sponsors on behalf of Regeneration Canada. Thank you to Gouvernement du Québec, Englobe, General Mills, the A&W Regenerative Agriculture Initiatives Fund. This event would not have been possible without your support, so thank you. So I'd like to introduce our guest now, Joel Williams. Joel is an independent plant and soil health educator and presenter on soil dynamics, plant nutrition, and integrated approaches to agroecological farming. Joel has worked throughout Australia and Europe and is currently based in Canada. He has a passion for teaching and sharing both scientific and practical information on soil health and agroecology, and has lectured to farming audiences internationally. So thank you so much for joining us, Joel. It's really an honor to have you, and have a great session, everyone.
Cool. Thank you very much. I'll just share my screen here. Okay, is that working?
Yes, it works.
Okay, cool. Okay, thank you very much. Pleasure to be here, and thanks for having me in this virtual event, and hi to everybody from hopefully all over the world tuning in. Pleasure to be here. So the topic for today is soil organic matter, and really the flow of the course today is that I want to cover three critical areas, three kind of major points of discussion, which will really firstly cover some of the, my slide's not working here. Oops, sorry, hold on, I've got to get my slide to work. There we go, okay. First, to cover some of the new ideas, the emerging paradigm around soil organic matter formation. So there's been a whole host of research in the last decade or so, and there's some new ideas emerging around the best ways to build soil organic matter and the nature of soil organic matter. So then after that, that will be the more dense part of the lecture. Then we'll move into some strategies to build organic matter based on some of those emerging ideas. And we'll kind of round that off with a discussion at the end about how do we use soil health and soil organic matter as a central aspect of soil health as a good foundation to build a system redesign, transition production systems towards regenerative and agroecological type approaches. Okay, so let's get right into it, because this first section will be probably pretty dense, so I've got to keep on pace. So it starts with, say, slides like this one, and there'll be all sorts of slides, images similar that we could put up on screen, highlighting that obviously in the last 50 to 100 years, especially since the Green Revolution, that we've seen yield increases consistently in a whole range of different areas and regions and crop types. Now, sure, certainly in just the more recent times, we're beginning to see that plateau, most certainly. However, but more historically, we've seen these quite significant increases in yield. And so we might assume then, well, with those increases in primary productivity, surely we might assume that our soil organic matters have also increased. If our yields have gone up, why hasn't our soil organic matter also gone up if a greater proportion of residues and crop residues and material is being integrated into the soil? Now, unfortunately, these things are not linear, and we've seen with quite the opposite trend with the intensification of agriculture. With these yield increases, we've actually seen declines in soil organic matter, generally speaking. There are exceptions to that. When we began producing in very arid and desert regions, and we brought water, into those arid regions, well, sure, that we overcame a moisture limitation there, and you can increase in certain examples there. But across the board, we would say that soil organic matters levels have declined with that intensification, despite yields going up. And that really comes down to the fact that a huge proportion of those crop residues and that biomaterial, that actually it is lost from the system. It is respired off. It can be oxidized off and also respired off as CO2. And it's estimated as much as 75% of residues can indeed be lost from the system. So straight away, we're then left with a smaller percentage of material that will and can become integrated into the soil. So plant compounds in various states of decay and the microorganisms that consume those compounds that all then overall make a contribution to building soil organic matter. So that bottom piece there is what we're going to really explore in more detail as we move through the presentation now. So only a small fraction is ever integrated into the soil. And there's a little bit of detail, a little bit of devil in the detail when we think about some of those yield increases, those historic yield increases that we've seen. Here we're looking at a study looking at, again, over time, differences in the modern wheat varieties. And what we see is that although we've seen increases in the above ground biomass, although we've seen increases in yield, these two bars here and here, and therefore overall increases in productivity, actually that came with a decline in the root biomass. You can see that around about that Green Revolution time, we've seen that the root biomass actually declined. And we see that similarly over here, above ground biomass has increased, but below ground biomass has declined. And that was crop breeding. You know, we bred varieties to be high yielding, and that's a trade-off. When plants partition more carbon to above ground, it's a trade-off. They're putting less down into the roots. So those yield gains, those biomass gains, they have been above ground, not necessarily below ground. And here's a nice image illustrating exactly that. Here we see a traditional variety of wheat on the left-hand side, an einkorn wheat versus a modern durum wheat on the left-hand, einkorn on the left and a modern durum on the right. Now, this is just a simple little pot experiment, you know, but nonetheless, same soil, same growing conditions. You can see that the traditional einkorn there, it allocates a lot more carbon down to those roots. We can see much greater root biomass down below. So genetics has been a factor that has contributed to less carbon allocation below ground in the last 50 plus years. The other factor is nitrogen and fertilization practices. And here's a study that looked at, as we increase nitrogen or fertilization intensity, so as we move from the left to the right there, you can see that as we increase fertilization practices, indeed we do increase the biomass. This is shown on corn and wheat. And although that fertilizer does indeed help us to increase yields, it does so by increasing, again, above ground biomass. If we look at the root biomass of these two comparisons, you can see that actually the root biomass is much the same of the two. The impact, the influence of the fertilizer, it drove more above ground biomass production. Yeah, and this is now a chart highlighting the same thing. This chart here is, this is from a very comprehensive review on below ground carbon allocation in plants. And this image here is a summary of 3 separate studies. And so again, what we're looking at is on the bottom x-axis here, as we increase nitrogen fertilization, we see that the amount of carbon that the plants allocate to the below ground, down to the roots, that the below ground allocation is declining with that increase in nitrogen fertilization. So it appears then that these are two key drivers, genetics and fertilization, that have helped us to drive above ground gains, and that has come at a cost to the below ground root biomass production. Now, that's very important because it is roots and not shoots that are the most important driver of building soil organic matter. It is roots that build organic matter, soil organic matter, not so much the shoots. Now, the shoots do make a contribution, yes they do, but they make a much smaller contribution. And it's a less efficient way to build soil organic matter. Roots is the more efficient way, and roots make a much greater contribution. So if the goal is to build soil organic matter, then really we need to be putting roots at the top of our priorities in terms of that strategy moving forward. So we have a growing body of evidence that supports this. Here's a selection of studies here that are all pointing in this direction, that it is root material, root litter, that makes this more important contribution than shoot material. And some of these studies are from forest systems, grasslands, and also croplands. There's a real mixed selection there in these systems, where the trend and the pattern is always the same. It's roots, not shoots, that are more important in building that soil organic matter. So I'm going to just cherry pick out one of these studies here, the Jackson one at the bottom there, and I'm choosing this one because it is a review study. So this is now a graph from that study, and you can see that it's reviewing a whole host of other studies. And what we're trying to do here is just look at these 2 main columns here. We're looking at what was the percentage of below ground carbon inputs that became soil organic matter, and what was the percentage of above ground, those shoots, that became soil organic matter. And if you just scan your eyes down these 2 columns, you can see here's the percentages of root material that becomes soil organic matter versus what percentage of the shoot material becomes soil organic matter. And you can see that consistently, it is the roots that make a much greater a contribution to building soil organic matter. So that's a really nice summary, a table there that illustrates that nice and clearly. So from that study, just highlighting one point here, they suggest that it is the root inputs are approximately 5 times more likely than an equivalent mass of above ground litter to be stabilized as soil organic matter. Okay, so roots, you know, on average, perhaps 5 or so times more important. Okay. So that means if we think about this from a plant production point of view, from a crop point of view, certainly from a cover crop point of view, the critical take-home message is that we've got to shift our attention to below ground. Here we're looking at a cover crop this is a photo from Scotland, again quite northern, you know, short growing season before the winter closes in. And we're looking at a cover crop here. This is the same field, 3 photos taken the same day. Now, the photo on the middle and the left, you might look at this cover crop and think, well, it's not a very big, impressive, showy cover crop. It might only be a few inches tall there. It's not a particularly huge amount of biomass above the ground. And we might look at that cover crop and think, well, was it really worth it? Is it really providing much benefit? Well, the key message is that don't judge a cover crop by what you see above the ground. We have to go below. And so again, here we have over on the right-hand side. Now let's look at the root systems. And well, I look at this photo on the right-hand side and I see a very healthy, good-looking root system there to me. I look at this cover crop and feel that it is bringing benefit. It is bringing functionality to that soil because of that root biomass, even though, okay, the above ground biomass may not be quite as impressive. Now, sure, we would all agree we would be much happier if this cover crop was 2 or 3 times bigger. You know, of course, if we had more shoots above ground, we're going to have even more roots below ground. Okay, so of course that's also true, but nonetheless, I would argue that this cover crop is still bringing great benefit. We know that plants prioritize root development early on before they prioritize shoot development. So in the early stages of plant development, of plant growth, they're focusing on roots. So even small plants like these, as we see in these images, they still have good root systems. And I look at that image on the right, and I think that's looking fantastic in terms of that root biomass and the potential contribution that that could make towards building soil organic matter. So it comes back to then, of course, photosynthesis. Plants have to breathe in carbon dioxide from the air, and they use that carbon as a building block to build their bodies, to grow those shoots and to grow those roots, that shoot and root biomass. It is this beautiful photosynthetic process that we have to thank for that. And the point then just goes a little further to say that plants do not just breathe carbon in, they also release carbon. And they release carbon through those root exudates, carbon compounds, sugars, carbohydrates, amino acids, proteins, all sorts of carbon compounds that they will actually leak or excrete out of those roots. And, you know, typically we talk about root exudates as feeding soil microorganisms, feeding the biology around that soil, around those root systems in order to, for example, cycle nutrients and deliver those nutrients to the plant. They're trading, the plant trades with the microbes carbon for nutrients. So indeed, plants do also give away carbon. They don't just breathe it in. And so the question becomes, well, what about those root exudates? What kind of a contribution do they make in terms of building soil organic matter? Okay, and it turns out that root exudates have been something or an aspect of soil organic matter studies that has been somewhat neglected. And fair enough, root exudates are very difficult to study. They are exuded from the plant. They may only live in the soil for a few seconds, a few minutes, maybe if they're lucky, before they are consumed by the microorganisms growing around those root systems. So they're very difficult to study. But they may make an important contribution to soil organic matter building as well. And this is a study here that's asked that exact question. Okay, if roots and shoots, if roots are more important than shoots, what about these root exudates? What about these rhizodeposits? And as the study here highlights, this study is called Evidence for the Primacy of Living Root Inputs, these rhizodeposits, not root or shoot litter. in forming soil organic carbon. So as it highlights here, recent theory suggests that those living root inputs, that they exert a disproportionate influence on soil organic carbon formation. But few field studies have explicitly testing this by separately tracking the living root inputs versus those litter inputs as they move through the soil food web and into those carbon pools. So here they show that those living root inputs, the exudates and the rhizo deposits, that they are 2 to 13 times more efficient than plant litter. That's the roots or shoots. They are 2 to 13 times more efficient than litter inputs in forming both slow cycling, kind of stable soil organic matter, as well as some of the more fast cycling fractions as well. And we're going to come back and talk about those two in a minute. So it appears that perhaps this is just one study. We definitely need more studies to build more evidence on top of this, most certainly. But this is one study that is pointing in a direction that says, well, okay, those root exudates may indeed be a very important piece of the puzzle. And there's a very important reason as to why this is, which we will now kind of tease out. And it comes down to something called carbon use efficiency. If we think about microorganisms growing in the soil, living in the soil, they are looking for carbon, they're carbon hungry. And when microbes are, for example, on the left here, given some root exudates, those root exudates, those simpler carbon compounds, those amino acids, etc., those are highly available. They are highly digestible, very easy for the microbes to assimilate, to feed on. So they can rapidly consume with ease those carbon sources. Versus our crop residues. Here over on the right-hand side, we have some mature plant litter here. You can see it would be rich in cellulose, in lignin. It has a very wide carbon to nitrogen ratio. It's very complex structural plant materials. This carbon over on the right-hand side, this is very difficult to degrade. It takes a lot of effort, a lot of energy to break apart, to break down those complex structural compounds to then digest them, versus those exudates on the left, which are highly available, highly labile, are easily assimilated. So I have a very complex technical image here to explain how this one works. So here we have a microorganism here, and over on the right-hand side is some small, simple food sources, again, that the microbe can easily digest. It can easily absorb these smaller carbon compounds, and it will then use those as building blocks. It will use those as energy to grow its body, to build its cell walls, to build all sorts of other structural compounds that the microbe itself needs. So it takes those smaller little pieces and it uses various enzymes that help to convert that, stitch them together to form these larger, more complex kind of structural things that the microbe itself needs, its cell walls, for example. So that's how the microbes will ingest their food. Now, if we present them with a very complex material, like these structural plant compounds, rich in lignin or cellulose, for example, well, the microbe cannot just simply ingest that. It cannot just take in that complex molecule. Microbes can take in some more complex molecules, but not this complex. So consequently, the microorganism has to break that down and turn that into these smaller components. So therefore, what the microbe will do is will synthesize these extracellular enzymes. Okay, and these are special molecules that will attack that complex plant litter. Okay, they will attack that and they will break it down, break it into smaller component parts, break it into smaller pieces. So then that complex material is broken down into smaller, more easily digestible, easily available carbon compounds, which then the microbe can ingest. So the point then to highlight here is that the production of these extracellular enzymes that the microbes have to release to break down the complex stuff, these enzymes come at a metabolic cost. The microbe has to expend energy. It has to waste energy in synthesizing these enzymes in order to break down the complex material to then in order to feed and digest. So that means that those microorganisms are less efficient feeders. They're having to waste that energy. If they didn't have to produce all these extracellular enzymes that come at an energetic or metabolic cost, well, they would have more energy, more resources to simply feed on more food sources and therefore grow more microbial biomass more efficiently. So this comes really down to the efficiencies of those food sources and the efficiencies of how easily microbes can take a food source and convert it into microbial biomass. And the more easy the food source is to digest, the more microbial biomass they can grow. The more complex the food source, the more difficult that is. They have to waste all that various energy. So that's really the process. It comes down to this question of efficiencies. And here we're comparing then, for example, things like root exudates here being highly available, and these structural plant compounds, lignin, cellulose, which are much more difficult to degrade. Now, this point about use efficiencies and how efficiently we can grow microbial biomass becomes very important, because part of the emerging paradigm on soil organic matter suggests that actually it is the microbial biomass, and particularly the dead microbial biomass, and this is called microbial necromass, that it is the microbial biomass and the necromass that makes a much more important contribution to soil organic matter than we previously also thought. And so this was a study looking at that, as the title suggests, soil organic matter genesis, microbial biomass as a significant source. The point is to say that the plant materials will come into the soil, the microbes will feed and digest and break that down, but at the point where they begin to run out of food and they begin to starve and they begin to die, it is this process of death, of decay, it is the microbial necromass that then is a key contributor or key builder of soil organic matter. It is the dead microbial bodies, it is that microbial necromass that then sticks to soil particles, interacts with soil minerals, interacts with soil aggregates into the soil physical surfaces, and it is those cell walls and those decaying microbial bodies that then interact in a dynamic process with the soil mineral surfaces to form soil organic matter. And so the study is suggesting that those dead microbial bodies make a much bigger contribution than we had previously thought. So that's a study from 2012. A more recent study just from last year, we were now here trying to quantify, well, how much microbial necromass, how important is it in terms of soil organic matter? And here the authors show that that microbial necromass can make up to more than half of soil organic carbon. Hence, we suggest that next generation field management requires promoting microbial biomass formation and necromass preservation in order to maintain healthy soils, ecosystems, and climate. Okay, so that's really quite significant, maybe up to 50%. Soil organic carbon is made up of these dead microbial bodies, the microbial necromass. And as highlighted also over here, perhaps it suggests that the fungal bodies, the dead fungal bodies, may make a greater contribution than the dead bacterial bodies. So the fungal to bacterial influence potentially having a bit of a factor there as well. Okay, so dead microbes, soil death, soil decay is a really important aspect of this. Everyone loves to talk about soil biology and soil life. We're all very excited about soil life and the concept of living soils. Here we are at the Living Soils Symposium. I would say that, in fact, soil death and soil decay, now I know that's not quite as sexy as soil life and living soils, but actually soil death and soil decay is of equal importance. It should be right up there in our consciousness, in our thinking of living soils and building soil organic matter. It is those death and decay processes that are also critically important. So let's kind of stitch all of these points together now to kind of bring some of these this discussion together, thinking about the two images here on the left that we've already discussed. What we then really have is two key pathways in which we can form soil organic matter. And the first one is the one we touched on about these extracellular enzymes, where the microbes will be releasing these enzymes which externally, outside of the microbial body, they attack the organic matter. They break it down, chop it up into smaller pieces. Now, as those get smaller and smaller, some of those carbon compounds, yes, can become very available, so very labile, very soluble, very available. But some of those may be still more complex. Some of those they may not enter a microorganism. They're just being broken down externally. They can still contribute to soil organic matter stabilization, mainly through physical processes. If some of these structural compounds that are not fully digested, not incorporated into a living organism, if those structural compounds get wrapped up within an aggregate, stabilized within a soil aggregate, well, then indeed they can become part of the stable carbon pool. But some of the other organic materials that do become more available to the microbes become quite labile, as well as the root exudates, as we discussed. These highly available fractions of carbon, they can be consumed by the microorganisms. And this study here was calling this the microbial carbon pump, the MCP. Once plant materials enter the living, go from dead carbon into living carbon into the microbial biomass. Once the carbon enters the microbial biomass, these fractions of the carbon are much more likely to become much more stable. And that's through those microbial necromasses we've touched on, and through some of the microbial metabolites. Microbes excrete all sorts of byproducts and waste substances, which are also more stable. So the point is to say there's two pathways, one that goes through microorganisms, one that doesn't go through a microorganism, but gets trapped and embedded within aggregates in order to stabilize it. So from that then emerges this these two new terms, which are part of this new paradigm in soil organic matter dynamics, what we call particulate organic matter and minerally associated organic matter. And so particulate organic matter is made from those complex plant residues, high in lignin, high in cellulose, etc. This is the enzymatic decay. This is not going through a microbial body. So this is the parts of organic materials that are just getting broken down into smaller pieces. So it really is a continuum of organic molecules ranging from complex all the way through to smaller and less complex. It's not that the food sources are one or the other. It's not simple or complex. It is a continuum of various states of decay. And so all of these fractions, these are called particulate organic matter. And this fraction of organic matter, these are less stable. They can live in the soil for maybe 1 to 50 years. It depends on a range of factors there, but these are considered the less stable forms. Now, if we had better quality litters, so for example, root exudates or younger, fresh green material, smaller carbon to nitrogen ratio, legumes, for example, well, this higher quality carbon, well, it is more available to the microbes because of those carbon use efficiencies that we talked about. So the microbes can more easily digest better quality litters. And therefore, once that carbon goes through the microbial body, forming biomass, which can then form necromass, that this then can attach and stick to the soil mineral surfaces. And it is that interaction of dead microbial body and interaction with the mineral surfaces of the soil that form this minerally associated organic matter. And this is the fraction of organic matter that is much more stable, okay? So up to 10 to up to 1,000 years. So the minerally associated organic matter is the one that we clearly want. I mean, we want both, and they both play a role. It's not to say one's more important than the other, but if we're talking more longer term stabilization, indeed it is this minerally associated organic matter that is more important in that context. And we know that therefore, better quality residues, better quality litter, lower carbon to nitrogen ratio, that's going to feed biology more efficiently than the lower quality residues with a high, wide carbon to nitrogen ratio. Okay, and again, that's just to illustrate that the physical processes are very important. It's the aggregation of soils is very important. When we aggregate soil and we create these lovely structures here, when the soil particles get stuck together and we form this aggregate, of course, is a ball. It's a sphere. It's not a two-dimensional structure like on screen. It is a ball. And all of the organic materials, organic carbon embedded within that ball, within that aggregate, can be captured, can be stabilized and protected from those microbial enzymes, protected from oxidation, protected from, therefore, loss. So any forms of soil organic materials, be that particulate organic matter or the minerally associated, any can be wrapped up within aggregates. Okay, and if it's wrapped up within those aggregates, well, it is stable. However, if that aggregate breaks down, well, then it is that particulate organic matter that is going to be more likely lost than the minerally associated organic matter, which is more stable because of that chemical, physical interaction that we see there. So aggregates do play a very important role in also capturing and protecting soil organic matter from oxidative losses or from microbial attack. And this was a big review study that really summarized that exact point. As the title here says, soil organic matter turnover is governed by accessibility, not recalcitrance. Okay, so the old model of soil organic matter formation was dominated in thinking that we wanted more recalcitrant carbon compounds. We wanted more difficult compounds to degrade, things like wider carbon to nitrogen ratio, cellulose, lignins. We thought that's what we wanted because those have a slower rate of decay. And therefore, the slower the decay, the more time that they are residing in the soil, bringing the benefits. Okay, but actually, so we had this very focus on the nature of the plant materials, the chemical makeup of those plant materials. But as we now know, it is the physical processes that are much more important than the plant chemistry. It's how well organic materials are physically trapped within those aggregates, or how well they are attached and adhered to those mineral surfaces, those physical processes that are much more important, not the nature of the plant material. So this slide, these last two slides will then kind of summarize this first half of the lecture. So this is, here we're looking at the old model of soil organic formation. We thought that root exudates, root inputs, those highly available fractions of carbon inputs, we thought that they did not make a contribution to soil organic matter, that they were too labile, they were too available. And it was bacteria that would rapidly feed on those, and bacteria would also rapidly respire a lot of that carbon off as CO2. So we thought, well, no, these were too simple. They weren't complex enough to be stabilized in the soil. So we didn't think that they made a very little contribution to soil organic matter. So therefore, the dominant narrative became, well, we want more complex materials. So this is focus on litter, on stubble retention, on more complex materials. We want more of them in the system. And it is fungi who will digest those, because fungi are better at digesting more of those complex materials. So we had this over-focus that this was the pathway to build soil organic matter. It had to be the complex stuff. Fungi were the organisms who were better at digesting that. And then as the fungi degrade and break that down, this leads to persistent organic matter, all these various humic substances. So now what we are thinking is turning that quite on its head. And this is perhaps the new model of thinking that, well, actually, soluble carbon compounds like root exudates, good high quality litters, these indeed can feed fungi and bacteria. It's not that it's one or the other. Fungi will happily feed on root exudates. It's a highly efficient source of carbon for them to grow their bodies. It's easier for the fungi to feed on exudates than it is to feed on this complex stuff that they have to, again, synthesize those extracellular enzymes, expend that metabolic energy to break that down. Again, it comes at a cost. So we know that fungi will quite happily feed on labile forms of carbon, as will bacteria. And then as those bacterial bodies and those fungal bodies die and form that necromass, well, both of those make a very important contribution to building soil organic matter. And we also know that, of course, that even root exudates, if they are trapped within an aggregate, if they are embedded within an aggregate, even small carbon compounds, so structurally simple carbon compounds, they also can be stabilized in soil organic matter pools through that physical stabilization, through being embedded and bound within those aggregates. Even simple stuff can certainly be captured and stabilized as well. So this is kind of the new model that essentially puts greater focus over to the more labile, the more available fractions of carbon versus the more recalcitrant and more persistent fractions of soil organic matter. So what I'll do here is just, this is now the first slide of the next section. I just want to quickly mention the points here to say that this is the transition where we will move into practical. We're going to talk to some of what is the practical implications of this. And I just want to preempt some of your questions. So just so you know that these are the types of things that will be coming in part 2. So just trying to prevent an overlap of questions here. So we'll talk about the roles of cash crops. Okay, how do we use this knowledge to manage cash crops better? And that becomes thinking about roots, not shoots, okay, using cash crops with bigger root biomass or higher root to shoot ratios. The obvious thing there is using perennials. They play a key role here, and maybe breeding new varieties with greater below ground traits. We can use various fertilizers or inputs to actually enhance root biomass. So I'll give some examples of that. There are various kind of biostimulants and input related substances that can enhance root development. Then we move into some of the kind of soil health principles, maintaining a living root. So keeping living roots in the soil for as long as possible, cover crops, relay intercropping, avoiding bare fallows, minimizing soil disturbance, so preserving those soil aggregates, of course, so minimizing soil disturbance accordingly. And we'll touch on some things around diversity, a little bit on polycultures. How do we use a variety of plant species for different rooting characteristics? Can we use companions and intercrops for their more so their roots, for their below ground benefits? We'll talk about cover crops, again, selecting cover crops with better rooting characteristics. And we'll touch on a few things around livestock integration and how we can use animals as part of this conversation as well. So that's the things that will come up in part 2, but that is now the end of the technical part of the lecture. So happy to now take some questions.
Great, thank you. Thank you, Joel, you did such a great job explaining a complex topic. So we do have quite a few questions here. We can go through a couple of them, and whichever we don't have time to answer right away, we can maybe get to at the end of the session. So we have one popular question here at the top of the list. So this is from Audrey from Gaspésie. She says, "Hi, Joel. Where does compost fit between particulate organic matter and the more stable, slow cycling one?"
Okay, it is a combination of both. So the benefit of a composting process is that, of course, you are using microorganisms to digest the plant material. So in that sense, when the organisms decay the compost ingredients, the feedstock of that compost, any of that feedstock carbon that is integrated into the body of the organism, into the living microbial biomass, then it is more likely to be stabilized as that dead microbial necromass as minerally associated. Whereas in a compost, of course, you will still have other fractions of plant materials that are in various states of decay in that continuum. So you'll have bits of particulate organic matter in a compost, and you would have more of those higher quality or dead microbial body fractions. Now, one of the best, if I can quickly add, one of the best strategies to then improve the quality of the compost or improve the minerally associated synthesis is to put a mineral component as a feedstock into the compost pile. So by that, I mean add some mineral soil or add some clay, and there you're then adding those mineral surfaces in which those dead microbial bodies can adhere to and become more of that minerally associated and stabilized fractions. So if you can put some clay or soil into your compost, that would be a key strategy that would help and improve the quality of it.
Okay. The second most popular question we got, what could be the contribution of fresh animal manure to soil organic matter formation? Also in relation to plant litter, root exudates, meaning in the context of grazing to promote root growth.
Yeah. Okay. So I will touch on a little bit of these in the next section. I would just, in summary, I would say the critical point here is that, as I mentioned, it's roots, not shoots, that help to build soil organic matter. So therefore, and I should stress before I go here, it's not to belittle the importance of shoots. You know, shoots on the surface, surface stubbles and litter protection on the stubble retention is critically important for soil health. You know, you want that stubble retention, particularly in dryland climates. You want that for moisture conservation, erosion protection, wind protection, habitat for insects, etc., etc. So shoots, I have to stress, shoots still play a critically important role here. Just they play a less important role in terms of building soil organic matter. But of course, they help with the efficiencies of everything else that's going on below the ground. So the point then to answer that question is to say that if shoots play less of an important direct role in building soil organic matter, I think that one of the best ways to use those shoots is to put them through an animal, because then you're going to let the microbes in the rumen, or the right microbes in the digestive system of the animal, to do that breaking down. So then the manure comes out, you're taking a low quality carbon resource that's low quality, not as efficient and good for the microbes. Let the microbes in the gut do the work. And then when you spit out that manure out the other end of the animal, well then, yes, you have changed the quality of that organic matter. It's now higher in nitrogen, it's now better quality, that the microbes in the soil can more easily digest and incorporate into their bodies. So I would argue that livestock do play a very important role in taking that inefficient carbon source and making it, of the shoots there I'm referring to, and making it more efficient and making it a better contributor to soil organic matter. And we'll come back to a little more details on that and your part about root exudates. Yeah.
Great. Okay, thank you. So next question is from Viboda. So we're saying, thanks for a fascinating presentation. And what role would sulfur and nitrogen play in the picture of building soil organic matter?
Yes, okay. Yeah, there's lots more to discuss in all of this topic. Of course, we only have limited time. But yes, you're right. Soil organic matter is not just carbon. Soil organic matter is made up of carbon and hydrogen and oxygen and nitrogen and phosphorus and sulfur and those other nutrients. So you need those nutrients also to help. Microbes need those nutrients too. So you need the nutrients there in order to help build that soil organic matter as well. So yes, we do know that nutrients play a very, very important role. Just in the same way that we know that plants need all of the macro and micro minerals, essential macro and micro minerals to grow their bodies, well, microbes need all of the essential minerals to grow their bodies too. So yes, nitrogen plays a role, and yes, sulfur and phosphorus as well. So under nutrient deprived systems, nutrient depleted ecosystems, yes, it becomes a limitation. It's harder to build that soil organic matter if you have those nutrient deficiencies there. And just comparing the minerally associated and particulate organic matters here, we typically see then that under those more nutrient deprived scenarios, you will see more particulate organic matter formation, because particulate organic matter is made up of lower quality, less nutrient rich materials, whereas the minerally associated fractions contain more nitrogen, contain more nutrients. So nutrients are very, very important in that picture. Yeah.
Thank you very much. Just a little reminder for all our participants. Let's keep the chat for any technical question, and keep all your content related question to Q&A. That way we can keep them, people can vote in it. We'll make sure we don't miss any, because the chat is going really fast, and I might not be able to scroll back to the top. There is a question here from Jael. Hi Joel, please could you explain the difference between oxidation and respiration of above ground plant material as they break down?
Okay, so that's part of the reason why shoots are a less effective way of building soil organic matter than roots. It's partly just very simply their physical location. The fact that they sit on the surface, that they sit on the above ground, that's one of the key reasons that they are then more oxidized and therefore lost versus the roots, which simply due to their physical location of being down in the soil, not on the soil, but in the soil, where those microbes are, where then that carbon can be broken down and converted into soil organic matter. So the difference between the two, between oxidation and respiration, is that the oxidation is just a chemical process. Respiration is a biological process. So oxidation is very simply where that oxygen and the carbon, there's no biological interaction that takes place. The oxygen will attack the carbon and break it down, and they will form CO2. Where then that CO2 is lost. So it's just a chemical process, chemical oxidation. And that's different from respiration, which is then a biological process where it is the microorganism, for example, in this example, who are then actually feeding and ingesting the carbon. And it's, I guess we could kind of call it a little bit like breathing. The microorganism is kind of breathing. They're breathing out that CO2 a little bit like we do. So in the process of feeding and digesting and expending metabolic energy, they are releasing some of that CO2. So you would say that at least with respiration, you're gaining some microbial biomass. With oxidation, that just chemical process, you're just simply losing carbon. But at least you're gaining some kind of functionality through the biological and microbial interaction when it's being digested and CO2 is being produced. So I would summarize to say one's a chemical process, one is a biological process.
Okay, great. Thanks, Joel. I see that we do have a lot more questions coming in, but I think maybe you can continue your presentation and then we can see which questions we can get to at the end. And again, if you'd like to see some of the questions answered, I invite you to click on the thumbs up under the question and we'll get to those quicker. So you can go ahead and continue your presentation, Joel.
Okay. Okay, so I should stress it's not this part where we will look at the how do we now manage for soil organic matter. This is not necessarily intended to be a fully comprehensive discussion on strategies to build soil organic matter. For example, there was that great question there about compost. I'm not particularly going to talk about compost here. This list of topics here are linked to the new paradigm of soil organic matter formation. I'm just trying to touch on how do we interpret some of that evidence and translate that into field conditions. So by no means is this a comprehensive list. It's just some of the points of discussion that are important for the first half of the lecture. So, and this second half, I'm going to show a lot more photos and images. We're going to keep this a little more fast-paced, a little less technical, a little more practical. So it should flow a little better for you all. And we're going to touch on some of these points here. How do we use cash crops to do better, to grow more soil organic matter? What fertilizers or inputs can we use? How do we apply some of the soil health strategies, maintaining living roots, minimizing soil disturbances, this transition around polycultures, diversity, cover crops, and some words on livestock. And as I say, all of that we will then link into some broader discussions about agroecology. So the first thing to say is a transition to perennials is a first and very easy step. Perennials allocate more carbon to below ground. They grow bigger, deeper root systems. So that's going to be a first and obvious and easy way, transition to perennials. But as I touched on, we could also be choosing varieties, current varieties, and certainly breeding varieties for the future of annual plants that do allocate more carbon down to the below ground, who do build bigger root systems. So there's the genetics factor there, perennials, and choose cash crops that have a higher shoot to root ratio, okay, so more carbon allocation to below ground. Then we could think about some of those inputs. This is a fantastic image of some of wheat and some wheat rhizosheaths, and you can see this fantastic root system here. This wheat plant was planted into a diverse cover crop that was all frost sensitive, so all of those plants then would have frosted off, and the only thing this wheat was planted with was molasses and humic acid. So, you know, certain things like nutrients, biostimulants, biofertilizers, even on current crops or current varieties, they can enhance this rooting behavior, the rooting biomass. So, okay, things, for example, things like phosphorus. Okay, we know that is a very important nutrient for root development. Sulfur, very important for root development in legumes. Boron, very important for root development in brassicas. So there are some nutritional things that we could think about and how to manage our current crops. And then things like biostimulants, I mean, seaweed extracts, particularly seaweeds that have a higher auxin content. It's auxins that stimulate more root development. And we know a range of things like various microbes, biofertilizers, they may play a role there in helping to stimulate these root processes, root branching, and more root development through some of these microbial interactions. So there's a range of kind of input-related strategies in which could be used as part of this discussion. I'm not really going to go into that detail. We're going to focus more on some of the structural system redesign strategies for this particular presentation, but I acknowledge that there's a big discussion there to be had around inputs and particularly biostimulants and inducing greater rooting responses in crops. And this touches on the question on livestock. Here we're looking at root exudation patterns and those rhizo deposits in annual crops, this is the pink, versus perennials in the green, so grassland in this example. And so you can see over time, we see annual crops, they have a rapid peak in their root exudation processes, and then it progressively declines. And that's because annual plants, they move into reproduction, they prioritize carbon allocation above ground, they prioritize carbon allocation to the developing seeds and grains and fruit. So the point is to say that the root exudation process, the bulk of the root exudation process happens in that first 4 to 6 to 8 weeks of an annual crop. So that's your critical time where you get maximum root exudation, very little thereafter. And that's different from a perennial, for example, here we can see this grassland where we also then have a nice peak, but we have a much more sustained release, a sustained decline of the rhizo deposition under more grasslands or perennial type plants. Okay, so again, that point about transition to perennials, they allocate more carbon below ground and they create a more sustained release of that carbon versus, say, for example, annual crop production. And this is why then the idea of things like, for example, grazing cover crops can be a very good example, particularly plants that will respond or recover from that grazing. If you give those plants that first 50, 60 days of plant growth, well, right at this peak here, if you were to come in and graze at that time, rather than letting the plant go to reproduction and turn off the root exudates, if you were to graze right at that time, well, you will set back the plant into that vegetative state again, where it will keep the exudates turned on. So the bulk of root exudation happens in the vegetative stages, and it slows right down in the reproductive stages. That's the key point there. So keeping plants in a vegetative state, grazing one great strategy to do that, or for example, mowing or some other defoliation in a cropping scenario, that can also help to keep the plants in a vegetative state. That keeps the root exudates turned on. So here we're looking at cover crops. This is just a nice example of a autumn time cover crop where we're keeping the soil covered, we're extending the growing season. Okay, the cash crop has been harvested. Now we are keeping roots in the ground, root exudates being pumped into the ground. We're extending that for as late as possible into the fall, into the autumn, before everything perhaps may get frost off or go into a deeper, into a state of dormancy there. So plus we have a nice picture of diversity here, keeping the soil covered with diversity, but critically extending the growing season, keeping living plants in the soil for longer. We can also do that then with things like relay intercropping. You know, right as these wheat plants are moving into a reproductive mode, they're turning the root exudates off. We then have the beans underneath, who are still very much in a vegetative stage of growth. They are still at that maximum stage of root exudation down below. Of course, this wheat would have been planted in the previous fall and the previous autumn. So we had a living root in the autumn, okay, a dormancy through the winter. Then that wheat kicks off in the start of the growing season, and we then relay a new cash crop underneath that with the beans. Again, we're just extending the season of the living roots in the ground, and a really good example, and factoring in that dynamic between plants in the reproductive state versus plants in the vegetative state. Another example here, this was a clover that was undersown into this oat crop, and so here's a great example of no fallows. You know, we're going through with the combine, we've immediately got living cover underneath the ground, on the soil. We've got living plants, living roots, those root exudates working for us. We also have a nice example here of diversity with a legume and a cereal. We also have a nice example of minimizing soil disturbance. You know, this is an organic farmer. He's undersown that clover, so he doesn't have to now do any soil cultivations to establish that clover after harvest. It's already there growing in the understory. And now that we open up the canopy after harvest, that clover will proliferate rapidly. So again, really nice examples of extending the living root season, minimizing soil disturbance, and bringing in some higher quality litters, the legume litters that you can see there. And then, okay, we can all acknowledge that if we could transition to then no-till or minimizing soil disturbances, we don't want to break apart those aggregates. Okay, so any strategy to minimize soil disturbance is a good one. Maintaining those aggregates to keep carbon trapped within those aggregates. So yes, go no-till if you can. A good middle ground, a good transitional strategy, maybe something like a strip till, as we could see here. So at least we're not cultivating the entire field, only a fraction of that field is being cultivated for the next cash crop, but at least we've maintained undisturbed soil in those inter-row areas. So again, a great transitional strategy for middle ground approach to minimizing those soil disturbances. Key message from this slide is don't judge your cover crop by what you see above ground. Okay, get out there and go dig up the soil, dig up the roots, and have a look at what is going on below the ground. That's how you should be assessing the success of your cover crop, is by what the roots are doing, not necessarily so much by what the shoots are doing. Now, again, I acknowledge there's a relationship there, and generally the more shoots, the more roots, but again, the key point is assess what's going on below ground. Not just above. Okay, and here's an example of an intercrop. We can see a pea with a canola, with an oilseed rape here. And this is a nice example of, again, a bit more diversity, but also we're changing the litter quality. So, you know, those peas, those are legumes, they're going to have a lower carbon to nitrogen ratio. This would be a good example of peas if it was combined with a cereal, for example. Again, that cereal, as we touched on, is a lower quality litter source. The legumes having a lower carbon to nitrogen ratio, they are a better quality to form, for microbes to digest and form that minerally associated organic matter. So even with this simple step from 1 to 2 into an intercropping situation where we have a legume and another cash crop here, we're bringing a better quality carbon material to help build that more stable formation. Now, we could also choose another companion, just as an example here, with bigger rooting characteristics. So the whole idea of companion crops, I think you could have one as your main cash crop for its above ground, for its yield, for your economic return, and maybe choose a companion for its roots. Can we align a companion with a big root system to bring more of those benefits of sequestering soil organic matter, building, stabilizing soil organic matter? Okay, then we have livestock, and this is just an example, as I touched on before. I think taking shoots, which are the least efficient way to build soil organic matter, and putting them through a pre-digestion, putting them through the animal, is a great way to take that low quality carbon source and turn it into a high quality carbon source in the form of manure. So I think livestock integration may play a really important role in this discussion. It makes shoots more efficient at building soil organic matter, which they typically are not. Okay, that's why I think livestock do have a really important role. Here's an interesting study that was comparing a grazed pasture versus a mowed pasture. So they took the grassland and they split it in half. On one half, they let the animals in and let the animals graze. On the other half, there was no animals, and they used machinery to mow the grass. Now, they found that where you excluded the livestock, the grazing exclusion, that this was associated with less carbon allocated below ground, less root biomass production, less root exudate release, therefore less microbial biomass that grew on those exudates and those rhizo deposits, and therefore overall lower soil organic carbon than where we just mowed. Okay, so there is an impact and influence of the animal. The animal itself does bring benefits to this process of carbon allocation to below ground, and therefore soil organic matter formation. So, and that's a big discussion, of course, around livestock, but there's lots of other studies that show that they do help to build soil organic matter. And then things like the grazing strategy. This was perhaps a little bit of the question we had earlier as well. We do know that when you don't graze quite so intensively, so you know the whole concept around, say, mob grazing, when you're only grazing, say for example, half of the shoot biomass, well, we know that that does not impact the root health or the root growth or the root recovery. It improves the root recovery there and the plant recovery when we graze less. As soon as we graze heavier, graze tighter, we see that actually roots become inactive. Roots stop growing. We see less root focus there. So again, the whole concept around mob grazing makes a lot of sense to me in the sense that it's very clear that when you don't graze as much of the tops, you maintain better, more root activity below ground. And I actually think that that's the key reason that those mob grazing styles appear to help build soil organic matter more effectively. I know strictly speaking, the academic evidence on this is not substantial yet. It's still thin. There are pieces of evidence out there that support this for sure, but there are many critics of the technique in the scientific literature anyway. If you ask a farmer, practically, farmers who are implementing such strategies are very supportive of it. The evidence, I don't think, has quite caught up yet. But my feeling as to why that grazing technique helps to build soil organic matter, it's mainly because we have less of an impact on roots. Many would say that it's because you leave some residue on the above ground and the animals trample that residue in, and it's that residue that helps to build soil organic matter. I think it's the fact that we've grazed less, we've had less of an impact on root slow down on root recovery, so the roots were able to maintain greater activity, and that's actually what's building soil organic matter. That would be my suspicion, that the roots play, again, a more important role than the shoots that's left behind with that grazing technique. That's just my opinion. Okay, so that's some of the kind of practical thoughts around how we might integrate some of that new science, new evidence. Let's then kind of bring it all together, thinking about system redesign. How do we transition then our production systems to a more regenerative or agroecological approach? And I'm a big fan of the ESR framework, the ESR kind of model of thinking about how to step through that transition to change production systems from input intensive through to more input independent and more agroecological approaches. And I'm a big fan of the system. I think it really breaks it down nicely. And it breaks into these three stages: the efficiency, substitution, and redesign. And the efficiency stage starts with the idea of, well, we're currently in an input-dependent system. Where we need to start is by improving those input efficiencies. We need to use less and less fertilizers. We need to use less and less pesticides. When we are using them, we need to use them with the utmost efficiency, and that means we can use less and less of them. And of course, the less and less of those we can use, we're opening the potential, we're opening the door there for the soil itself to perform those functions, particularly the soil biota, to perform some of those functions. So we're dialing down fertilizer use, we're dialing down pesticide use, only spraying pesticides when absolutely necessary, not calendar spraying, use them only when critically required according to thresholds, but also improving input efficiencies, foliar applications of nutrients, for example, more efficient than soil applied, seed treatments more efficient than soil applied, integrated nutrient management, carbon-based fertilizers. There's all sorts of strategies that we can use to help improve the efficiency and make those gradual steps down on using a little bit less and less and less inputs by becoming more and more and more efficient. Okay, so that's critical because as we wind those down, we're opening the door for the soil biology to become more functional, more active. So the next stage would say, okay, I've got my inputs down. Now I want to substitute out of them. Now I don't want to use them at all. So how do I substitute away from those? And that's where we would then have those discussions around organic fertilizers, biological inputs, biofertilizers, more natural, more botanical pesticides, for example. So one example might be, again, we're going to cut down our nitrogen fertilizer, then we're going to try and use more nitrogen fixing bacteria. So using biofertilizers and inoculants, we're going to substitute out of the fertilizer into a biological fertilizer. We're going to substitute out of the pesticide into a botanical pesticide, into a biopesticide, these kinds of things. So that's your next phase where we're now removing those inputs, which generally many would agree are detrimental to the soil health, to soil functioning, and we substitute those into these softer options. And that's your substitution phase. And really those two processes then, those two stages really pave the way for the third part, which is the system redesign. This is where we then redesign the production system to not need to use any of those inputs or minimal of those inputs. And we design with an ecological framework. We look into ecological systems, we study ecological systems, and we apply that learning to agriculture. And that's essentially what agroecology is. It's about mimicking the landscape, trying to design, use the natural synergies and efficiencies of ecosystems into the design of the production system. So that really means, as I said, mimicking the landscape, but particularly designing ecological parts into the farm, integrating ecology into agronomy. And really, therefore, simply what we're doing is creating multiple functions. We don't want our land to simply produce food. We want it to produce food and support ecosystem services or ecosystem functions at the same time. And that's really what it comes down to. We want to have a multifunctional process. And so this is a really good stepwise transition. I should stress, it's not that you would do 1, 2, 3, or you do only efficiency, then you only do substitution, then you only do redesign. It's the lines between those are blurred. You can begin them all at the same time, but that's just a way to think about that process of transition. Okay, so, and here's a really nice image, couple of images illustrating why we need the redesign, why that's so important. On the left-hand side, you can see 2 images here. These are biologically simplified landscapes. We have a vast areas of monoculture. There's not a great deal of diversity in the landscape. It's a biologically simplified landscape. We have one big main cash crop on the top, and okay, we see lots of other crops down in the second picture. And this is in clear contrast to the right-hand side, where we have more elements of the ecosystem designed in, these semi-natural parts of the farm system also designed into the picture. And this is really important because let's picture these fields on the left. This, for example, these 2 fields on the left, they could in theory, for example, be organic fields. We could have actually gone through the efficiency and the substitution phase. These might be through there. We've increased efficiencies, we've reduced inputs, we've substituted out of those inputs using more biologicals. We're still locked into an input dependent system with E and S. This could be organic system, but hey, it's still a monoculture. It's still a vast area of where is the ecology, where is the diversity that's designed into that picture? And that's why efficiency and substitution are critically important stages, but they're not enough. You have to bring in redesign. And it is the redesign that then brings in the multifunctional elements, and particularly the landscape scale. It's not just about in-field diversity. It's also about what is happening across the landscape and using natural or semi-natural parts of the landscape in order to help improve our production. Yeah, so that's really what redesign is all about, is kind of bringing these elements into the picture. And the core of that idea simply says we want the production system to be multifunctional. Natural ecosystems, they're very good at providing ecosystem services, all of these kinds of things over here, but they're not so good in terms of feeding us, in terms of their productive element. An intensive cropland, of course, it's better at producing and feeding, but they are less effective at providing these other ecosystem services. So what we simply want to transition towards is a multifunctional agro-ecosystem. We want to produce food and support ecology at the same time. And that's the kind of framework or thinking in which we could apply. And I really love this image because if we think about historically the divide between organic and conventional farmers, you know, there's been classic dichotomy, this classic kind of disagreement there on you're on one side or the other. And it's become a very, not a productive way to think about moving forward. And I really like this image because it brings a third piece into the puzzle. It says bringing in ecology, bringing in ecological thinking. Now, on the left-hand side is the industrial model. On the right-hand side is your more organic type model. The reality is now we have things like regenerative agriculture, which are probably somewhere in the middle. And the point is to say it really doesn't matter where you are on this spectrum. You might be an industrial farm, you might be a smaller scale organic farm, you might be something in the middle. It actually doesn't really matter. All of those production systems would benefit if they designed more ecology into them, if they got more diverse, more mixed farming systems. If we design more ecology into an industrial system, it would improve. If we design more into an organic system, it would improve. If we design more ecology into a regenerative system, it would also improve. And I think that's a much more useful and functional way to think about that process of system redesign. And so I would encourage all farmers across all spectrums to just think about more ecological principles. So how do we do that? The most easiest and simple way is increase plant species diversity. This is a critical and easy first step that will start that transition. So we can increase species diversity through, using cash crops, more novel cash crops or wider rotations. That would also be a great way to increase diversity. Or then talking about things like more annuals, more perennials. If you're a perennial system, bring some annuals in. If you're an annual system, bring some perennials in. The use of summer or winter crops would also play a role. And then, as we've discussed, things like cover crops, things like companion crops, things like intercrops, bringing more diversity in field. Again, very important first step. And that's, again, what kind of regenerative agriculture is about. It's bringing more of that diversity in field. And where agroecology is good is that it thinks about bringing more diversity in a landscape scale, not just in field, but also a landscape scale. And that's also very important. So that could be then things like those biodiversity strips, field margins, agroforestry, silver pastures, bringing animals, trees and grasses all together, many different strategies. But the key message is plant species diversity is a great way to increase that transition. So we could use things like cover crops. Again, I'm just going to show a few nice images to illustrate that. Here's some nice diverse cocktail cover crops, bringing different plants with different functionalities that can benefit that system. Here we have a grassland, a pasture scenario. On the left-hand side is a 12 species mix. On the right-hand side is an 18 species mix. And so we can see that more diversity is obviously bringing a little bit of a synergy there. It looks like we have greater biomass production on the right-hand side where there's a more diverse pasture species in that scenario. What about, again, this intercropping discussion, even going from 1 to 2? 1 to 2 can actually make a big difference. Here we have some maize that was undersown simply with some ryegrass. I mean, soil protection, living cover, living roots, root exudates, a little bit of diversity. Even that step from 1 to 2 brings a lot of benefits. And we can really see that clearly in terms of soil protection with this image, quite a stark example of some of those benefits. And then we bring in some of the more ecological things, so pollinator strips, flower strips, these kinds of things that can help to draw in the beneficials and the insect, the predators and the pollinators, these kinds of things. Another example here of using Phacelia as a nice strip through a cash crop, again providing pollen and space for the beneficial insects. One more example of that here from North America. These are the prairie strips. So again, these are kind of a mixture of perennials and grasses and flowers and plants, which are permanent covers, perennial strips there in between the cash cropping. And again, why not trees, agroforestry, and livestock? And all of them together, grazing livestock in woodlands, you know, using trees, using animals, I think also is an interesting space, which there's definitely more opportunity and more potential there, bringing trees and cattle together, which I think is just, for me, conjures really nice imagery. You know, it's in all of the debate around climate change and it's let one or the other, you know, trees will save us, animals are evil. I love this idea of bringing animals and trees together and highlighting that things can really work synergistically together as well. And again, just a really nice image there highlighting the benefits of diversity, particularly if we think about below ground, you know, choosing deeper rooted species, choosing deeper rooted varieties, encouraging more root biomass. That's going to be great for building soil organic matter, but that's also going to help us be more efficient with the nutrients that are in the soil, help us use less fertilizers, use less inputs, because we will be leveraging the existing fertility that's already in the soil down at depth and accessing that with those deep root systems and bringing that up and cycling that. A quick study on the benefits of plant diversity on soil organic matter. So here's a study, a global meta-analysis, so a big review study by analyzing 1,001 paired observations of plant mixtures and corresponding monocultures from 121 publications. We show that both soil organic carbon content and stocks are on average 5 to 8% higher in species mixtures than in monocultures. These positive effects increase over time and are more pronounced in deeper parts of the soil. These species mixture effects are consistent across forest, grassland, and cropland systems and are independent of background climates. Okay, so that's a very big overall review study, 1,001 observations highlighting that more diverse mixtures of plant species mixtures helps also to build soil organic matter. So soil biodiversity, we could argue that it is the natural capital of farming, and it is soil organic carbon that is the currency of that capital. Soil organic matter carbon definitely directly influences soil life, the biological functioning, therefore the soil fertility, nutrient availability, and therefore the overall yields, while also driving soil health. And that is also the foundation of the wider ecosystem functionality as well. Okay, so I want to share one last study with you. I'm very close to the end now. I just want to share one last study that is a really good example of where ecology and production can both win. You know, we have this classic narrative that that when we produce food, that it comes at a cost to the ecosystem, a cost to the landscape. And this is a really nice example of a real win-win where we can design production systems to be productive, but also to support ecology. And this is a study out of the UK. And what they simply did is take the field here that you can see on the right, and they simply reclaimed the field margins. So they cut into the field margins and reclaimed them and planted up the field margins with tall grass species, a mixture of 4 grass species that they planted up around those field margins, and gave the field margins back to the ecosystem and focused on the production inside the field, but gave the field margins back to the ecology. So there were 2 treatments here. They cut into the field, they reclaimed those field margins, first by 3%, so they took 3% of the productive land out of production and gave it to these strips, to the field margin strips, and then they cut deeper into the field and took 8% of the land out of production, giving it to the ecology, planting up with these tall grass species in the field margins. So of course, as we cut into the field, as we lower the area under production, So we're growing on less area of land. Of course, yield is going to go down, isn't it? Because we're now growing on less land, because we're giving away land to the ecosystem and planting that up to provide habitat for the beneficial insects. So what did they find when they did this to the study? These first two charts on the left, we're looking at some of the yield indices here. So you can see that the first one is business as usual. That's our control. That's our yield index there. The middle column here, column B, this is where we have taken 3% of the land out of production and given it to the ecosystem. And you can see that we saw an increase in the overall yield, even though we're growing on less land. And when we cut into that field again, even deeper, we gave 8% of the field land over to the ecosystem. This is column C. You can see that actually we have a further increase in yield again. So even though we were growing on 8% less area here in this treatment, the overall yield, the productivity in that less area was greater. And that's very simply because those field margins, those ecosystem biodiversity strips, they provided an ecosystem service, a habitat for beneficials to then migrate into the cash cropping area, where they then performed a function and helped with integrated pest management. So over here on the right-hand side, that's what we're looking at. We're looking at the predators. We're looking at the predators and the crop pollinators, the beneficials. And you can see here that particularly when we gave away the 8% of the land is where we saw the biggest increases in beneficials, in pollinators and predators, etc. Not so much in the middle column in this instance, but we certainly saw greatest increases in the beneficials in this third treatment, where we gave 8% of the land over to the ecology. Okay, so this just summarizes a really nice example where indeed we can increase yield and we can increase ecosystem functionality at the same time. Okay, there are examples out there, but they have to be designed for. I acknowledge that not all of them pan this way, so we need to understand those interactions and design the production systems accordingly to support these kinds of win-wins. Okay, so in conclusion, efficiency and substitution, that ES part of the transition, that is key to reducing the dependency on those inputs. That is a key first step. Okay, we've got to start using less of those inputs. And in order to transition through those two stages, through the efficiency and the substitution phase, of course, a healthy and functional soil is essential. You won't be able to reduce your inputs. You won't be able to reduce that dependency on inputs unless you have a healthy and functional soil. And so therefore that opens the discussion into soil organic matter. You could argue that soil organic matter is probably the most important aspect of a healthy soil, however we define that. Now, there are exceptions to that, but broadly we could, for the most part, we would agree that soil organic matter is probably one of the most, if not the most important aspect of a healthy soil. So that's going to form the foundation of creating that healthy soil to reduce those dependency on inputs. Okay, and that's the discussion we had today, the role of roots, root exudates, microbial necromass being really important, aggregate preservation, keeping the soil covered, minimizing soil disturbance, all of those things that we discussed in part 1 of the lecture. And these then pave the way to system redesign. Bring more plant species diversity into the system. This is a key strategic starting point. Okay, then we need to apply a little broader holistic or systems thinking at that landscape scale. Okay, it's not just at a field scale. We need to widen that to the landscape scale, and that's where we've got to bring some ecological thinking into the picture, and therefore design more of that ecology into the agronomy. Like that example that we just looked at from the UK, bringing ecological areas, habitat, be that through strips, be that through field margins, we have to design and integrate more ecology into our agronomy. Okay, thank you very much.
Great, thank you, Joel.
I see that we're right on 5:30, so I apologize, we've gone a little over time. Hopefully people can stick around for a few questions.
That's all right. Clearly it shows that you're very passionate about this subject, and I think a lot of our participants are as well. We have many, many questions. We could probably be here for another hour, but we just have a couple minutes. So we'll start with the questions that were voted for the most. And you might want to read this one with me, Joel. It's a little bit longer, and I'll still read it out loud, but it's the first one at the top of the list. So from Hendrik. Who says, there are studies that challenge the notion that increasing soil carbon on the top layer of soil, example with no-till, actually results in a net increase of carbon in the soil. Instead, that it's mostly a redistribution of carbon from as much as 1 meter deep with no net overall increase. Are we confident today that increasing soil organic matter through agricultural practices is really effective in increasing the total amount of carbon sequestered, removed from the atmosphere? And then there's an example of a study from the USDA.
Yeah, sure. Yeah, you're right that a lot of the early thinking around no-till suggested that it was a very important strategy to build soil organic matter. And it is a piece of the puzzle, undoubtedly it is. But as you correctly highlight, what no-till does is concentrate that organic matter to the more surface horizons. Yes, but really that's referring to, yes, the distribution of that soil carbon. Yes, it's concentrated at the surface, perhaps less in the deeper parts. The answer to the question is yes, we can still help to sequester soil organic matter. And as the weakness of the purely no-till approach, as you're highlighting, the weakness of it is that it stops the no-till, it stops the disturbance, it stops the aggregation. That's good, but that's one piece of the puzzle. As well as no-till, we have to design more diversity and soil cover. And these are principles of, say, conservation agriculture. It's not just about being no-till, it's about keeping the soil covered with residue or living plants, as we kind of discussed today, and designing more diversity. And it is when you design more diversity into the system, particularly with the deeper rooted crops, as we were just talking about, that is where you will get greater carbon sequestration, carbon being taken out of the air and deposited at those deeper levels. So yes, we can still improve soil organic matter levels in either no-till or tillage systems. There are different strategies either way, but yes, it can happen at depth, and you need deeper rooting plants is the key to that for sure. So even though no-till will concentrate at the top, if they bring in a more conservation ag approach, get more diversity, more deeper rooting, you'll open up more opportunities for that.
Do we have time for another question, Sarah?
Yeah, I think we can do one last one. It's a really good one too.
So this one is maybe related to your last slide, maybe. And if I can resume it, how can we speak to farmer in a way that appeals to them, that make really change how to make the narrative of, oh, competition between my cover crops and my cash crop, for example? And this question was from Brooke Hayes here.
Yeah, I think it's, I think we are in the process right now of seeing that transition. There are many farmers waking up to ideas of regenerative agriculture, and therefore part of that is more plant species diversity. I think that is a process, that is a transition that we're going through right now. I agree with you, it's not all the way complete yet. We're not there yet. But from my experience in the interest in uptake in ideas around soil health that is happening in the last few years, in the last 5 years, the interest is exploding everywhere. I think we are going through the middle of that transition right now. And the point is to say that plant species can be competitive. Indeed, they can be, or they can be collaborative. It can go either way. And it's about then, and that's what, the practice of intercropping, I think why intercropping is such a central tool into breaking down that paradigm that you refer to, that paradigm that says any other plant is a bad plant. Intercropping, in my opinion, is going to be probably the biggest lever, the biggest driver of breaking that barrier, breaking that paradigm, because we're beginning to see lots of examples out there where even that transition from 1 to 2, from a monoculture to even just a biculture there with 2 plant species, that we begin to see all sorts of beneficial interactions under those intercropping systems, lower disease pressure, greater nutrient use efficiencies, greater yields. I mean, there's all sorts of benefits that are emerging out of intercropping. And I would argue that that's going to be the answer, because ask a farmer to transition to a very diverse system might feel overwhelming. Ask a farmer to transition from 1 to 2, that may seem a lot more manageable. And when they do that and see the benefits of just 2 species versus 1, they will be more curious as to know, well, what will happen with 3 and 5 and 10 and 15. So I feel that intercropping is going to be a critical tool in breaking that transition, breaking that paradigm.
Great. Well, thank you so much. And I want to be mindful of everyone's time. I see we're already over time here. And thank you all for your questions. And if you'd like to take another look at Joel's slides, we'll be putting those up on Whova on the app, so you'll be able to access them there. Thank you again, Joel, for a great presentation. So about Whova, if you haven't downloaded it already, we really encourage you to do so. You'll be able to access today's slides and most of the other presentations as well. It's also a great tool to connect with others, with speakers and other participants. And you can also schedule your own virtual meeting to keep the conversation going. So if you'd like to keep talking about, discussions that were had today as well, you can set up a virtual meetup, a group meetup, and continue that conversation. So finally, if you'd like the work we do at Regeneration Canada, and you'd like to support us by becoming a member, please visit our website at regenerationcanada.org to join the movement. We'd love to have you all in our network and to keep this conversation going. Just a reminder, tomorrow at 1 p.m. Eastern Time, we're going to get things kicked off with a workshop on defining regenerative agriculture and the debate about certification. And we invite you all to join us. We'd love to hear your perspective. So thank you so much, Joel, again for joining us. Did you have any last words to send people off with?
I was just seeing lots of people say thank you in the chat box there. So yeah, my pleasure. Thank you for tuning in. I hope it was a pretty dense and intense lecture there. Sorry I didn't have as much time there for questions. I perhaps went a little over time than what I wanted to, but I hope you all got something out of it, and thank you very much for tuning in. My pleasure to be here. Thanks for having me.
Great, thank you so much. Bye everyone, thank you for joining.
Okay, bye.
Bye.